Welcome to This Explainer. Today, we're diving into the structure of proteins. But, I want you to think about it a little bit differently. I want you to imagine a massive, microscopic construction project. Literally every single second, right inside your cells, biology is building these incredibly complex, fully functional 3D machines, and it's starting from nothing more than raw, simple bricks. It's an absolute masterpiece of architectural engineering. But just like any complex building out in the real world, if the structural integrity is compromised even a tiny bit, the results can be catastrophic. So we're going to trace this entire journey together, from the very first architectural blueprint all the way to the devastating consequences of a structural collapse. Here is our architectural blueprint for today's explainer: the amino acid blueprint, spirals and pleated sheets, building the 3D machine, the folding process, and finally, when architecture goes wrong. Let's get right into it.
Okay, let's dive into Section 1, the amino acid blueprint, the linear sequence that starts it all.
And it all starts with this crazy little number, 20. There are just 20 common amino acids found in proteins. That's it. It's absolutely wild to think about, but every single protein in your body, no matter how ridiculously complex it is built from a combination of just these 20 fundamental building blocks in this microscopic construction process.
But hey, bricks are pretty much useless without mortar, right? These amino acids are joined covalently by peptide bonds. Think of the peptide bond as an incredibly tough mortar. It has this partial double bond character, making it shorter than a typical single bond, and it is entirely rigid and planar. It's so tough, in fact, that it's highly resistant to conditions that would normally just destroy or denature proteins, things like extreme heating and high concentrations of urea. To actually break this non-enzymatically, you'd need prolonged exposure to a really strong acid or base at super elevated temperatures.
So you might be wondering, how do scientists actually read this linear blueprint? Well, they use a process called Edman degradation. Because a polypeptide chain is read from one end, specifically the free amino end, or N-terminal, scientists use a special reagent to label just that very first brick. It introduces a specific instability into just that first peptide bond, which allows it to be hydrolyzed and detached without cleaving the rest of the chain. You identify the piece you just chopped off, and then you just repeat the cycle. It's this brilliant, automated way to read the sequence step by step.
Moving on to Section 2, spirals and pleated sheets, framing the structure.
Now, what's really interesting here is how nature essentially uses two main architectural frames to build these structures. First up, the alpha helix. This is a tightly packed, coiled core, a right-handed spiral where the side chains extend outward so they don't get in each other's way. And it's stabilized by extensive hydrogen bonds that run parallel to the spiral. Now contrast that with the beta sheet. Unlike that tight helix, these are composed of segments of polypeptide chains that are almost fully extended, creating this broad, pleated surface. Here, the hydrogen bonds run perpendicular to the polypeptide backbone. So we've got two totally different ways to frame the interior of a protein, both utilizing hydrogen bonding to maximum effect. But of course, you can't just build in a straight line forever. You need corners, you need adjustments. Certain amino acids actually act as structural disruptors. Proline is a perfect example of this. Its unique geometry makes it act like a kinked pipe, completely disrupting a smooth alpha helix. Large numbers of charged or bulky amino acids will also repel each other and interfere with those spirals. Now, to keep the structure compact and globular, we have beta bends. These are basically 4 amino acid turns, often utilizing that kinked proline or a tiny glycine that safely reverse the direction of the chain. And when you start combining these helices, sheets, and bends, you get motifs. These are distinct, super-secondary geometric patterns that form the real core of the molecule.
Let's move to Section 3 and see how this builds. Building the 3D machine, assembling the domains. For those really large polypeptides, and we're talking usually over 200 amino acids long, the structure is divided into what we call domains. These are the fundamental, functional, and three-dimensional structural units of polypeptides. The cool thing is, they fold independently of one another. They act almost like independent, compact rooms or wings within our larger building, each with its own specific structural and functional characteristics.
Okay, but what actually holds this entire 3D tertiary structure together? Well, 4 crucial forces act as our ultimate structural supports. First up, disulfide bonds. These are actual covalent linkages between 2 cysteine residues. They are incredibly strong, and they basically prevent the protein from unfolding in the harsh extracellular environment. Second, hydrophobic interactions. Amino acids with nonpolar side chains absolutely hate water, so they hide in the interior of the protein, which drives that compact shape. Third, hydrogen bonds on the surface interact with water to enhance the protein's overall solubility. And fourth, we've got ionic interactions, or salt bridges, where negatively and positively charged groups attract each other. These four forces cooperate perfectly to stabilize the tertiary structure.
And just really briefly, we have to mention quaternary structure. This is when entirely separate polypeptide chains, known as subunits, unite to form massive cooperative functional complexes. A classic example of this is hemoglobin in your blood. It's a tetramer where the binding of oxygen to one subunit actually increases the affinity of the other subunits for oxygen. It's essentially just perfect molecular teamwork.
All right, Section 4, the folding process. Enter the construction foreman.
So we've talked a lot about the final structures, but the actual assembly, the folding itself, happens in just a matter of seconds to minutes within the cell. And it's driven heavily by that hydrophobic effect we mentioned earlier, where water is released as hydrophobic groups cluster together. But, this process really needs supervision. Enter the molecular chaperones, also known as heat shock proteins. I love to think of these chaperones as tiny construction foremen, or maybe cellular bouncers. Left to their own devices, exposed hydrophobic regions on an unfolded protein might stick to things they shouldn't, causing a really messy aggregation. Chaperones like HSP70 bind to these rowdy exposed regions to keep them in check. Others, like HSP60, actually form these stacked, cage-like macromolecular structures. The partially folded protein goes into the cage, folds up in a safe, isolated environment, and is then released when the building is finally, truly ready.
It's an incredibly elegant, highly orchestrated process. But what happens when folding fails?
Section 5, when architecture goes wrong. The devastating collapse.
When the cell's quality control system fails, misfolded proteins aren't properly degraded. Instead, they just accumulate. And when protein architecture collapses like this, it leads to some of the absolute most terrifying degenerative and neurological diseases known to medicine. Let's break down the mechanics of Alzheimer's disease for a second. We actually see two distinct architectural failures here. First, amyloid beta. This is a peptide that gets improperly cleaved and aggregates into a beta-pleated sheet conformation. These non-branching fibrils form massive, extracellular amyloid plaques in the brain that are highly neurotoxic. Second, we have the tau protein. Normally, tau helps assemble the microtubular structure of neurons, but in Alzheimer's, it becomes hyperphosphorylated and insoluble, creating intracellular neurofibrillary tangles that just block normal cell function. The architecture is literally being destroyed from both the outside and the inside of the neuron.
And then we have prion diseases, things like mad cow disease or Creutzfeldt-Jakob disease. So the crucial point to understand here is that we are looking at the exact same primary sequence blueprint, but with vastly different outcomes. Take a look at the normal cellular prion protein, PRPC, found on the surface of your neurons. It's composed mostly of alpha helices. But the infectious form, PRPSC, there are absolutely no primary structure differences here, literally no mutations in the sequence. But the 3D conformation has completely changed. A number of those alpha helices have been replaced by beta sheets. This single structural swap makes the protein form insoluble aggregates that are extremely resistant to degradation.
I want you to consider this striking quote from our source material. The infective agent is, thus, an altered version of a normal protein, which acts as a template for converting the normal protein to the pathogenic conformation. There is no virus here, no bacteria, no detectable nucleic acid at all. It is just a single, misfolded protein acting as a malicious template. When it encounters normal, healthy prion proteins, it literally forces their structural alpha helices to unravel and collapse into diseased beta sheets. It is a domino effect of pure architectural collapse, and it is invariably fatal.
From 20 simple amino acids to beautifully complex 3D machines, protein structure really is a marvel of microscopic engineering. But it leaves us with a pretty heavy final question. How does knowing that a single misfolded protein, just a mere shift from a spiral to a pleated sheet, can bring down an entire human brain, change the way you view the fragility of your own biology? It's a delicate, high-stakes balance happening inside you right this very second. Thank you so much for joining me on this explainer, and keep questioning the incredible structures that make life possible.